Hello, welcome to my talk. Today I'll be talking about the nasal and the oral cavities and how they form in the salamander. For some background on our interest with this research, craniofacial development is being studied because the cleft lip and/or palate is one of the most common birth defects in humans. Specifically in amniotes, which is the group consisting of mammals and reptiles, was studied. In amniotes, the region of the nose and the mouth form from three craniofacial prominences, illustrated here in the mouse in the red, purple, and green. The dashed line in the magnified image shows the fusion zone. These prominences come together and fuse in all groups of amniotes. This fusion allows for an intact oral and nasal cavity to form. If this fusion were to fail, then an individual would have a cleft lip and/or palate. We were interested in seeing the evolutionary history of these prominences and whether they arose in mammals and reptiles, or if it was an older structure that was shared with amphibians and fishes. During amnio-craniofacial development, the nasal and oral cavities develop as interconnected structures during the fusion of prominences. This connection is obviously retained in adult mammals, as illustrated by milk coming out of our noses when we laugh too hard while drinking it. This project aims to examine craniofacial prominences, specifically examining the evolutionary timeline to see when craniofacial prominences arose. As a representative amphibian, we decided to examine the aquatic salamander, known as the Mexican axolotl, for the presence of these craniofacial prominences. It is a very common lab research model and a known model of developmental biology. Before we get into data, I would like to show you some information to orient you. We decided to use micro-CT to analyze the morphology of the axolotl embryo. Here is a representative scan. First, here are the eyes. Here are the nasal cavities, and here is the mouth. We have outlined the developing oral and nasal cavities in order to assess their spatial relationship. You will see the mouth is in green, the left nasal cavity is in purple, and the right nasal cavity is in yellow. In terms of results, 6 stages were examined. In stages 11 and 13, we see that the oral and nasal cavities develop separately with some distance between each other. As you can see, there is a large gap present between the nasal and the oral cavities. This was surprising because we know in adult salamanders, the nasal and oral cavities are connected to each other, just as they are in mammals, including humans. To further elaborate on our results, in days 15, 17, and 19, the nasal and oral cavities progress an inch closer to each other until they eventually fuse. The oral cavity even goes underneath the nasal cavity, surpassing it. As we can see in day 19, the nasal and oral cavities eventually do connect and form a configuration similar to what we observe in mammals and reptiles. When we take a closer look at day 19, this CT scan shows the connection between the oral and the nasal cavities. There is now a similar connection and conformation to humans in other amniotes, where the nasal cavity opens into the oral cavity. Illustrated here is the narrow opening that connects the two structures. As a result, salamanders and amniotes have reached the same endpoint through different means, which is a fascinating phenomenon often seen in embryonic development. Ultimately, the face forms in a different manner, but we do achieve the goal of having the nasal and oral cavities connected. It is a different mechanism than in amniotes. There are no prominences observed in axolotls. Therefore, prominences are most likely an evolutionary novelty in amniotes. For future lab direction, we would like to go back into the lab and perform some histology to complement the micro-CT analyses. Moreover, we are interested in identifying the mechanism for the opening of the nasal and oral cavities through the cell proliferation and cell death analyses. Thank you for joining my talk today, and we hope you enjoyed it and learned something new and fascinating about axolotls.